The overall goal of the following experiment is to investigate the effect of the apoptotic stimulus Fas ligand in the developing cerebellum of wild-type and mutant mice. This is accomplished by first dissecting the brain of wild-type and knockout mice between postnatal days 8 and 12. The second step is to slice the brains in 400 micrometer thick slices and isolate the cerebellum from each slice. The third step is to culture the cerebellar slices for 5 days to make sure they are healthy before inducing cell death with Fas ligand. The final step of the procedure is to analyze the effect of Fas ligand in the different cerebellar cell populations of wild-type and knockout mice based on immunofluorescent staining for the apoptotic marker tunnel combined with cell-type specific markers like calbindin for Purkinje cells.
The main advantage of cerebellar slice cultures is that it allows the study of the cerebellum in its original structure, unlike primary dissociated cultures where the neuronal networks and cellular organizations are disrupted.
So this method will allow to answer some key questions in neurosciences, especially because one has the wild type and the mutant mouse, so one can study comparative development, one can study physiology and also electrophysiology and pharmacology.
Though this method can provide insights into the function of Fas ligand in cerebellar cell survival, it can also be used to study the effects of Fas ligand in the cortex and hippocampus.
Generally, people new to this method will struggle, because in order to obtain healthy cerebellar slices, it is critical to put them into culture as quickly as possible, without compromising their integrity. Therefore, it is important to have a good dissection and slicing skills before attempting the whole process.
Where possible, the steps of the following procedure should be performed in a tissue culture hood. Prior to dissection, prepare ACSF media for brain dissection and slice preparation, as well as slice culture media according to the written protocol, and store them at 4 degrees Celsius. On the day of dissection, prepare tissue culture plates for culturing the cerebellar slices. Add 1 milliliter of slice culture media to each well of a 6-well plate. Then, use sterile forceps to place the culture plate inserts into each well. Ensure that there are no air bubbles trapped beneath the insert. Place the culture plate in a tissue culture incubator set at 37 degrees Celsius and 5% carbon dioxide for at least 2 hours to precondition the media. Next, bubble the ice-cold ACSF media with carbogen gas. Saturation of the media with carbogen alters the pH of the solution and produces a color change from red to orange. Pour a little of the ice-cold bubbled ACSF media into a sterile petri dish and a 6-well plate. Place these on ice together with the plate of the vibratome that will be used for slicing the brain. The petri dish containing bubbled ice-cold ACSF media will be used for dissecting the brain from a P8 to P12 mouse pup. Following dissection, use a razor blade to make a sagittal cut in one hemisphere to provide a flat surface for mounting on the vibratome plate. The rostral part of the cortex may also be cut. Dry the chilled vibratome plate with a paper towel. Then, add a little superglue to the stage and spread it in a zigzag manner with the tip of the superglue tube to form a thin adhesive layer. Use a spatula to transfer the brain to the vibratome stage with the flattened side in contact with the superglue. Once the brain is attached to the vibratome plate, add enough ACSF media to cover the brain and add ice below the plate to keep it cold. Cut the brain into 400 micrometer slices. Then, use a plastic Pasteur pipette with a cut, widened end to transfer the slices to the 6-well plate containing ACSF media on ice. Working under the dissecting microscope, use 2 insulin syringes with small diameter 28 gauge needles to separate the cerebellum from the rest of the brain. If any of the slices have superglue on the edge, use the 2 syringe needles to remove the glue from the slice. Use the plastic Pasteur pipette with a cut widened end to transfer the slices onto the top of the filters, adding 1 to 3 slices per insert. Ensure that the slices are fully extended and that no liquid is covering them. If necessary, aspirate excess media with the pipette. Incubate at 37 degrees Celsius and 5% carbon dioxide for 48 hours. After 48 hours, replace half of the media volume with fresh media, making sure that no air bubbles are trapped underneath the insert. The remaining original medium contains slice-derived trophic factors that are important for cell survival. The apoptotic challenge is performed after 5 days of culture. Add the Fas agonistic antibody Jo2 to the cell culture media in the wells to be treated. Leave half of the wells untreated as controls. Once 24 hours have elapsed, aspirate the media from under the wells. Then, fix the slices by pipetting 1 ml of ice-cold 4% paraformaldehyde under the insert and 1 ml of PFA on top of the insert. Allow the slices to fix in the PFA for 10 minutes at room temperature. After 10 minutes, aspirate the PFA and wash briefly with 2 ml of cold PBS, pipetting 1 ml on top of the insert and 1 ml below the insert. Next, aspirate the PBS and pipette 1 ml of 20% ice-cold methanol in PBS underneath the insert and 1 ml above the insert. Incubate for 5 minutes at room temperature. Then, aspirate the 20% methanol from the well and wash the slice and well with PBS as before. Pipette 1 ml of 0.5% Triton X100 in PBS on top of the insert and another 1 ml underneath the insert. Incubate for a minimum of 12 hours at 4 degrees Celsius to permeabilize the slices. Following permeabilization, aspirate the 0.5% Triton X100 in PBS and pipette 1 ml of 20% BSA in PBS on top of the slice and 1 ml beneath the slice. Block the slices for a minimum of 4 hours at room temperature or overnight at 4 degrees Celsius. When blocking is complete, use clean scissors to carefully cut the piece of membrane insert attached to the cerebellar slice. Then, use clean forceps to transfer the piece of membrane into a 24-well plate containing PBS. Aspirate the PBS and add 250 microliters of tunnel reagent into each well, making sure that the reagent covers the entire surface of the slice. Incubate at 37 degrees Celsius for 1 hour in the dark. When the hour has elapsed, aspirate the tunnel reagent from the wells and replace with 500 microliters of PBS. Protect the plate from light and incubate at room temperature for 10 minutes to wash. Repeat this process twice. After the last wash, aspirate the PBS and add 250 microliters of a 1 to 1000 dilution of the calbindin antibody in PBS and incubate overnight at 4 degrees Celsius in the dark. The next day, aspirate the primary antibody solution and wash with PBS 3 times for 10 minutes. Then, add 250 microliters of secondary antibody diluted 1 to 500 in PBS. Cover the plate with aluminium foil to protect from light and incubate for at least 3 hours at room temperature. Following incubation, wash the slices 3 times for 10 minutes with PBS and, using mounting media that contains DAPI, mount the slices onto a glass microscope slide and cover slip. Image the slices with a confocal microscope using the 488 nanometer and 561 nanometer excitation wavelengths for calbindin and tunnel respectively. This image shows a healthy cerebellar slice after 2 days in vitro. The cell body layer appears translucent and the foliated structure of the cerebellum is clearly seen under the microscope. After 7 days in vitro, the foliated structure of the healthy cerebellar slice is still visible. In addition, dark cell bodies, probably macrophages, usually cover the slice. The confocal image seen here is representative of a fast-treated cerebellar slice. The Purkinje cells appear green after staining with a calbindin primary antibody followed by an Alexa 488 conjugated secondary antibody. The apoptotic cells positive for tunnel staining appear red, as the tunnel kit used is labelled with TMR red. Purkinje cells do not appear in a single row, but are clustered, forming a folium-like structure.
While attempting this procedure, it is important to remember to work in tissue culture hood whenever possible, and to minimize the amount of time the cerebellar tissue is exposed to possible contaminants like bacteria or fungi.
Using this procedure, other methods can be applied to these slices, including biochemistry and electrophysiological analyses, to provide additional information on protein content as well as neuronal excitability.
This technique was originally developed to study the effect of Fas-mediated cell death in Purkinje cells carrying mutations that affect the Fas pathway. Purkinje cells rarely survive in primary culture, so this procedure was the best experimental approach.
So I hope after watching this video, you will have a good idea as to how to make cerebellar slices and how one can study the apoptotic stimuli and its effect on different cerebellar cell types.